Hi, welcome back to Membranes. We finally get to the section on how does cellular transport actually work. Here we go. Passive transport is my number one. It's the movement driven by molecular motion, also called Brownian motion. Molecules are bumping into each other and they just tend to move around their environment. If it's warmer, they will move faster. If it's colder, they will move more slowly, but they will continue to move. So passive transport is going to be this movement of molecules that's driven just by the bouncing of those molecules into each other. Simple diffusion works this way. When water diffuses simply from one place to another, when potassium can just diffuse through that membrane, it's going to be simple diffusion. And facilitated diffusion is also moving molecules from a high concentration towards where there are fewer of them, towards a lower concentration, but it's going to involve one of those protein carriers, one of those proteins that are embedded in that membrane. So no energy, no ATP, it's still simple, but not right through the lipid bilayer, but with the use of a carrier. Osmosis is also movement of molecules from where they're more concentrated to where they're less concentrated, but in this case it's limited to water. The movement of water molecules from where they're in high concentration to where they're in lower concentration. We need to tell that apart from active transport that will require chemical energy, and that's usually ATP and all the systems we talk about in this class. So there's going to be active transport proper, where you need ATP transporting a molecule across a carrier, across that membrane. Or sometimes it'll be vesicular transport if it's not a carrier involved, but we actually blast right through the membrane. And that's going to be in the form of endocytosis and exocytosis. So there's no carrier. It's usually a molecule that's way bigger, and the only way it can get through that membrane is if it disrupts the membrane itself. It's kind of like a barrage on this wall. It's just sort of breaking the wall down that will reassemble back together again. So what are the requirements for simple diffusion? There needs to be that concentration gradient. There needs to be more molecules in one area and fewer in another. There needs to be that random molecular motion I've been talking about. And this is a really good example with the little orange balls. There's a difference in the number of solutes between the 2 points. There is a concentration gradient. It's more concentrated in solutes in area A and less concentrated in solutes in area B. Those molecules will bump into each other and move from region A to region B. Simple diffusion. No different from molecules in the air. Something smelly, a skunk sprays, you don't smell it at first, and then as those molecules bump into each other and disperse, you're now smelling it in your porch, coming in through your window. Until when? This will continue going from area A to area B until they're approximately evenly distributed. They will still not stop, they will continue to move, but they seemingly are evenly distributed. So we want to think about the properties of a particular molecule that's going to influence its diffusion across the cell membrane. Obviously, some of them do it more quickly and some of them do it more slowly. What is it about those molecules that's allowing them to go more quickly or more slowly? What is it? What are the properties of that molecule? And there are only two. One is size, and that makes total sense, right? If it's a larger molecule, it's going to be more difficult for it to cross that membrane. The second property is lipid solubility. Is it soluble in that lipid bilayer? Will it be able to cross that lipid bilayer? So if it is a lipid, likely will. If it's not, it likely won't. So the greater the lipid solubility, the greater the possibility of it crossing the membrane. We refer to this as polarity. Polarity has to do with charge. Nonpolar or no charge molecules can pass through the membrane by simple diffusion, because here we are talking about a fat. Fat's a nonpolar. We're also talking about smaller molecules like oxygen that can cross by simple diffusion. Carbon dioxide can cross the membrane. It's a good thing too, because this is how we're going to get oxygen, right, to our cells and eliminate carbon dioxide from our cells. And then the fat-soluble hormones, for example, like steroids, anything fat-soluble. Fat-soluble vitamins is the other example. Vitamin D, for example, is a fat-soluble vitamin. Vitamin C is a water-soluble vitamin. This is a very nice chart that exists in your book. All these diagrams do. Of the factors that influence the rate. So now we're going to think of not only does it cross and does it not, but how quickly. That's the rate of net diffusion of a substance across the membrane. So this is called Fick's law of diffusion. You do need to know which of these increase the rate and which of these decrease the rate. Don't worry too much about the formula down here that has square root of. Don't worry about that. Just which of these would be denominators, which would be above my line, think of it that way, and below, numerator and denominator. So, and they're pretty obvious. So once we go through them, I think it'll be clear to you. The first one, and we have arrows. As the concentration gradient of a substance increases, concentration gradient, so there's lots of these molecules in one area, very few in the other, big concentration gradient, then it is more likely that the rate of net diffusion is going to be high. It's going to go fast. Sorry. What about surface area of the membrane? So if we have membrane that's way larger, you have a greater surface area for more of these molecules to go through. It's kind of like a room with many doors or a room with only one door. That one with many doors is going to be able to have a faster rate of molecule movement. If it's lipid soluble, well, we just heard, right? If it's lipid soluble, it's going to be able to cross pretty quickly. If it's less lipid soluble, then it's going to have more trouble. So those three are going to be placed here above my line. Let's think of it that way, because the effect they have on the rate of diffusion is that they increase that. It's a direct relationship between the rate and those three variables. The other two variables we're going to then, they're going to be inversely related to the rate of diffusion. The higher the molecular weight of the substance, in other words, the bigger the substance, then that rate of diffusion is going to be lower. It's going to drop. And the distance that it has to travel. So if there's a lot of fluid somewhere and this molecule needs to transverse the membrane and then get past this fluid and then get to another cell, that's a greater distance, or it could be thickness of that membrane, the harder it is going to be for it to cross. So it's those 5 variables that are going to determine the rate, the speed at which this molecule can cross that membrane. So rate thought of sort of as distance over time. Now for osmosis, it's the diffusion of water. This always is confusing because when we think of solute molecules, we're not thinking of water, you're thinking of the salt, of the sugar. But the same goes for a water molecule, which is usually the solvent. If you have many water molecules in region A and fewer in region B, there's going to be a concentration gradient and water will go from where there's a lot of water to where there's less water. So let me read this to you now. Osmosis is the diffusion of water from high concentration of water to low concentration of water. If I switched that and showed it to you in terms of solute, it's flipped. You would say water moves from low solute concentration to high solute concentration. What's that mean? It's the classic example of you putting salt on your lettuce. So the solute then is the salt on the outside of the lettuce. What happens to the lettuce? If you leave it there long enough and don't eat it and it's got salt on it, it's going to wilt. Why? Because water moved from where there was less solute, less salt inside the lettuce, to where there was high salt outside the lettuce. And those cells all lost their water. So they went from where there was a lot of water to less water, but they also went from where there was low solute to high solute. So salt really pulls water out of places. And you know that, right? You get beef and you put water on it, and you can totally dehydrate it. And that's how we preserve. That's how, before refrigeration, things were preserved, right? You removed the water. So we say solutes suck. They suck the water out of things. So because of osmosis, the body compartments can be in that osmotic equilibrium. Remember again, I'll say it in red, not individual solute equilibrium. So the osmotic equilibrium is maintained at about 300 milliosmoles. It's a measurement of osmotic pressure. We're not going to look at that number again until we get to the kidney, which is probably the last week of this course. The easiest way to think of osmosis is in a U-tube like this, where you have region A and region B, and you have a membrane and water and a solute, in this case glucose. This membrane does not allow these glucose molecules to cross, but we need equilibrium. So if these molecules cannot cross the membrane, but water could, then water is going to move, think of it, from A to B or from B to A. So if water is attracted by the solute, it's going to go towards B where there was greater solute. So the solutes still are the same number on either side, but the concentrations now have equalized because water has moved by osmosis into the more concentrated solution. Osmotic pressure is the force that you need to apply to this side B here to oppose that osmosis that was happening. That is osmotic pressure, the pressure that must be applied to oppose osmosis. Let's stop there. Thanks for listening. We'll keep going soon.